we were discussing the molecular mimicry that is shown by the pathogenic antigens to the host antigens, where some similar epitopes of pathogenic antigens would raise antibodies in the host, and those antibodies will cross react with the self antigens, resulting in some abnormalities and some damage to the cells, which is an autoimmune response. There are many such examples. We'll discuss those examples. Let us see. One of them is cell wall M protein of streptococci. There are different strains in the streptococci. A, B, C strains are there, of which A group strain, there is a protein that is called as M protein. And that M protein, when it stimulates the antibody production in the host after the infection of streptococci, it would result in cross reactivity with the cardiac antigen, that is especially cardiac myosin, resulting in weakening of the muscle fibers of the heart, and that would result in heart failure in the person who gets infected by streptococci strain A or group A of the bacteria. Then another group of streptococci are known to cause in the children glomerulonephritis, where the antigen cross reacts with the basement membrane of the glomerulus of the kidney and resulting in the childhood itself the kidney failure. And that is also another example for the streptococci. And you have heard about Epstein-Barr virus. Epstein-Barr virus is a DNA virus. It is also known to cause cancer. Long-term residence of this virus is bad for the human health. It is also going to cause Burkitt's lymphoma, a lymphoma of the lower jaw near the salivary gland. And this virus produces a DNA polymerase for its replication, being a DNA virus. And that DNA polymerase, being a different protein in the body of the host, would initiate antibody production. And that antibody which is produced will cross react with myelin basic proteins of the myelin sheath. Myelin sheath is made up of some basic proteins, and they are in short form called as MBP, myelin basic proteins. And when the antibody produced against the Epstein-Barr DNA polymerase cross reacts with myelin basic protein, the person would suffer from demyelination. Demyelination is nothing but, you can imagine an electric wire is there which is insulated with a plastic or rubber coating from outside. The copper wire is insulated. You imagine that electric wire, you remove that insulation and conduct the electricity through it, there will be short circuited. Same thing would happen to the person who loses the myelin sheath. Each neuron and each conducting nerve is provided with protection of myelin sheath. And that myelin sheath acts as an insulating layer. And loss of insulation results in cross currents and short circuiting. Nerve conduction becomes very, very difficult in multiple sclerosis. It is debilitating autoimmune disorder. Then comes another mimic, that is the polio virus. When the person gets infected by polio virus, if he is not immunized against the polio by polio vaccination, the VP2 protein coat, that is the protein coat, VP2 protein in the protein coat of the polio virus, would initiate antibody production in the host. And that antibody will cross react with acetylcholine receptor, and it will bind to the acetylcholine receptor and makes it non-functional. And I had given this example, myasthenia gravis. It will result in myasthenia gravis, where voluntary movement of the person will be suppressed. Whatever he wishes to do, he will not be able to do. Those voluntary actions will be suppressed, because all voluntary actions in our body, what we want to do, will be done through acetylcholine transferase and the acetylcholine neurotransmitter. Okay, so acetylcholine receptor will be blocked because of the antibody. Let us see other examples. I had mentioned about heat shock proteins. Now there are bacteria that produce heat shock proteins on infection. And one of the bacteria is Mycobacterium tuberculosis, which causes the tuberculosis. And this produces a heat shock protein called as HSP60. And this HSP60 will be recognized as an antigen in the body and antibodies will be produced. And those antibodies will cross-react with the collagen in the cartilage. And by cross-reacting with the collagen in the cartilage, they would damage the cartilage. And generally, it would result in rheumatoid arthritis. And the rheumatoid arthritis is of the joints, where the cartilage that helps in capping the joints and lubricating the joints will be destroyed, resulting in arthritis. So that is another example of cross-reactivity of the pathogenic antigen with the host antigens. There is a disease called as ankylosing spondylitis. Moment spondylitis is mentioned, you know that it is concerned with the vertebral spine, the vertebral problem. Generally, people suffer from slip disc. There are cartilaginous discs that are acting as pads between the vertebral centra, helping the vertebral centra to absorb the shock of our walking. When we walk, the vibrations are transferred to our spine. Vertebral centra crushing against each other because of our body weight. They are protected by the cartilage padding. And that cartilage padding gets damaged in ankylosing spondylitis, means it is destroyed by the autoantibodies. And sometimes even the vertebral centra will fuse, making the person's mobility next to impossible. Vertebral column becomes stiff. And that is because the person who has inherited a haplotype for class 1 MHC, which is HLA-B27. Remember, when you had dealt with major histocompatibility complex genes, A, B, C genes belong to class 1 MHC. In that, HLA-B27 haplotype, which is one of the haplotype, if one inherits it, that haplotype antigenic epitopes are shared with a bacteria called as Klebsiella pneumoniae. The antigenic epitope of HLA-B27 is shared with Klebsiella pneumoniae. If the person gets infected by Klebsiella pneumoniae, and unfortunately he has a haplotype of HLA-B27, then the antibody produced against the Klebsiella pneumoniae will react with HLA-B27 epitopes and resulting in ankylosing spondylitis. And it is a debilitating disease. Nowadays, surgeries are available and one can do with artificial implantation of the disc. One can save the person of the fusion of the vertebra. But if it is not taken care, the person is unable to bend. His vertebra will become stiff like a steel rod and he will not be able to bend. So the reason being, the antigens of Klebsiella pneumoniae share the epitope with HLA-B27, which causes the disease called as ankylosing spondylitis. And some other examples, if we take, there are certain viruses like the viral antigenic epitopes. The type C retrovirus or paramyxovirus. When the antibody is produced against this virus, when it infects the human being, you can imagine those antibodies will react with even nucleoproteins. Nucleoproteins could be even RNA complexed with a protein in the cytoplasm of a person, and it is complexed with the spliceosomes or performing some functions in cytoplasm, and that RNA also is going to be destroyed. And there are reports of these antibodies getting transferred across the plasma membrane in the person suffering from type C induced autoimmune disorder, and those antibodies reaching the nucleus and damaging the nucleic acid in the nucleus, that is DNA with histone. Similarly, the antibodies that are produced against type C paramyxovirus would also react with red blood cells, or our RBCs, cross react. So one person who has been infected by this virus may suffer from an autoimmune disorder which is called as systemic lupus erythematosus. I will come to this disorder a little bit later. Why? Because it will be systemic, and all the damage that he is going to experience will be all over the body. Large number of different types of cells will be attacked because their nucleoproteins will be under attack, and the person will also suffer from deficiency of RBCs because of the cross reactivity of the antibody produced against type C retrovirus against these antigens. Then comes another virus called as reovirus. And this virus, when it induces the antibody production in a person, large number of endocrine glands will be affected, and that is called as polyendocrine disease. Like for example, the antibodies will cross react with pituitary, pancreas, gastric mucosa. The gastric cells are also endocrine. Some of the cells which produce certain hormones, gastrin and other hormones, are endocrine. Okay, it will also cross react with the nuclei and the glucagon producing the alpha cells of the pancreas, then the growth hormone and the insulin. Okay, so these hormones also will be deficient in the person because both these hormones, growth hormone and insulin, are peptide hormones. Glucagon is also a peptide hormone. So the antibodies will cross react with not only the endocrine glands, it will also prevent the endocrine secretions from acting. And that's why it is called as polyendocrine disease. There is a virus called as Coxsackie virus. And when the children get infected by this Coxsackie virus, in the early young stage itself, they will suffer from diabetes 1, type 1 diabetes, because of the antigenic epitopes of this virus shared with the beta cells, which are insulin producing cells in the pancreas, in the pancreatic islet. So those antibodies will go and destroy the beta cell, and due to destruction of beta cells, the child will suffer from insulin deficiency in the childhood itself and suffers type 1 diabetes. So these are the some examples of molecular mimicry, and these are common examples. That is why I have dealt with these common examples, because people, unfortunately enough, if they get infected by these viruses or the bacteria, they will suffer in the bargain with autoimmune disorder. Therefore, the physician who treats them against these viruses should take care that they will never suffer from autoimmune disorder. And nowadays, monoclonal antibody therapy is available. Moment the person is infected by these pathogens, the doctor can advise the person to take monoclonal antibody therapy to prevent the antibodies produced against these pathogens from cross-reacting with his host cells, antigens, and protect him from autoimmune disorder. It is possible. Nowadays, intervention is available. It is possible, but slightly it is costly, not affordable to the poor people or the middle class people. Anyway, the hospitals can help these people. Then hormonal factors also could induce autoimmune response, and major culprit is estrogen hormone. And that estrogen hormone in women, whenever it is in excess in the menopausal stage, where it is supposed to subside when a woman has reached the menopausal stage, the estrogen levels have to significantly reduce. But unfortunately, in some women, for some years after the menopausal stage is achieved, for some years that hormone will be still at a higher level. And when it is at a higher level, it is very, very bad for the woman, unfortunately, and very sad that woman has to suffer a lot. The one of the dangers caused by estrogen levels being high in menopausal women is it triggers the gene action and puts the gene of interferon gamma into on mode. Means interferon gamma will be produced continuously in that woman without any necessity. And interferon gamma is supposed to be produced only for the inflammatory response when a person gets infected by the pathogens to, in fact, the contain the infection or the destroy the pathogen through inflammatory response as well as ADCC response. But unfortunately, when there is no pathogenic infection and the woman has high levels of estrogen, it will keep the gamma levels high. And because of the interferon gamma levels high, that will act on the tissues, which will cause inflammatory swelling in joints and resulting in the arthritis. The women, majority of the women who reach menopausal stage or post-menopause suffer from the arthritis, which is in fact not even inherited. Just because of the estrogen level being high in her, she will suffer from arthritis, and that will result in rheumatoid arthritis later. Why? Because the rheumatoid factor will be produced, which will be in excess and it will keep accumulating in the joints, resulting in joint pain and destruction of the joints. Okay. Then the second most important thing about the estrogen is when it is in very high level, the incidence of breast cancer or ovarian cancer increase in women after the postmenopausal stage. And it is also observed that because of the estrogen level being high, some women suffer in the menopausal stage afterwards with systemic lupus erythematosus, which I just mentioned to you. And systemic lupus erythematosus is another detrimental disease where the antibodies are going to be produced against large number of different types of cells, and the person will suffer from the deficiency of those cells because of the destruction caused by the autoantibodies. This is as far as the factors that could cause or induce autoimmune response if the regulatory mechanism in the person fails of fine discrimination of self and the non-self, or if there is an imbalance in the hormonal levels, or if the infections by the pathogens take place and the pathogens mimic antigens of the host cell, then there is a possibility of autoimmune response. So coming to the different types of autoimmune response, let us classify them. They are classified into two major types. One of them is organ specific autoimmune disease or autoimmune response, organ specific, which will be confined to certain organs. Then systemic autoimmune response. The systemic autoimmune response is, it is not specific to any organ, it will be systemic, means all over the body, the systemic autoimmune response. We will first deal with the organ specific autoimmune response, which is further subclassified into two types. One, the disease, autoimmune disease mediated by destruction of the cells of the organ. Okay, that is mediated due to destruction of the cells of the organ. And the other one, disease mediated because of stimulating or blocking antibodies of a particular cells of the organ. And that will result finally in destruction of the organ. Initially, the antibodies, autoantibodies will be produced. They may stimulate the cells or they may block the cells. We'll come to that little later. First, we will discuss the autoimmune disease mediated by cellular destruction. First among them is Hashimoto's thyroiditis. Hashimoto's thyroiditis, if I have to discuss, first I have to show you the normal structure of the thyroid follicles. This is the section of the thyroid which is stained with hematoxylin and eosin. And the normal follicles look like this in a healthy person. It is, which is in fact providing single layered epithelial cells. Those are called as thyroid follicles. And in between the follicles, there is connective tissue matrix, which is extracellular matrix. And almost all thyroid follicles will contain within them thyroglobulin, which is an acidic protein. Therefore, it will stain with eosin and will be pinkish in color. And this is the normal architecture of thyroid in all individuals who have a normal thyroid functioning. And when Hashimoto's thyroiditis takes place, where the autoantibodies will be produced against different molecules in the thyroid. One is against the thyroglobulin, that is the thyroid hormone. Autoantibodies will be produced against the thyroid hormone, that is thyroglobulin. Autoantibodies may also be produced against thyroid peroxidase, which is an enzyme responsible for production of the thyroglobulin. Okay, production of the thyroglobulin. And the another possibility that the Hashimoto's thyroiditis may take place is because of the destruction of the thyroid epithelial cell antigens. Okay, thyroid epithelial cell antigens. So thyroid peroxidase or thyroglobulin or thyroid follicular antigens. Okay, thyroid follicle antigens. So these antibodies could be of any three different types, and these autoantibodies will react with thyroid, and the normal architecture of the thyroid, which is like this, will become like this, where the thyroid follicles will lose their architecture, the thyroglobulin secretion will decrease, and thyroid will be infiltrated by these blue small dots that you are seeing are nothing but the TH cells and the antigen presenting cells, that is macrophages. In fact, even neutrophils will be there. There will be large population of neutrophils, which are phagocytes, large population of macrophages, and large population of TH cells. They will infiltrate the thyroid and occupy the majority of the thyroid space. And it is as good as a kind of inflammatory response. And I had mentioned that DTH response, delayed type hypersensitivity, DTH response is brought about by TH cells. And this is as good as DTH response. The DTH response results in destruction of the thyroid, and autoantibodies will be responsible for that to trigger DTH response. And due to inflammatory action, the thyroid is destroyed and the person will suffer from hypothyroidism. Hypo means lower. Hypothyroidism. And I told you that hypothyroidism also leads to hypothyroidic goiter. There will be swelling in the thyroid gland on either side of your neck, and that swelling is because of the inflammatory swelling of the thyroid. The person will have deficiency of the thyroid hormone. The second important another disease which is caused as diabetes mellitus type 1 due to autoimmune response is because of destruction of the pancreatic islet cells, especially the beta cells. And this is the pancreatic islet. And in this, okay, and periphery to that, all these are the exocrine regions of the pancreas, which secrete the exocrine hormones of the pancreas. But this islet will secrete, because its islet is made up of alpha and beta cells. Alpha cells will secrete glucagon, beta cells will secrete insulin. And this Langerhans islet, which is called as pancreatic islet, will be destroyed by the autoantibody, especially that autoantibody is directed against the beta cells. And that antibody, autoantibody, is called as PICA, P-I-C-A, P-I-C-A in capital letters, pancreatic islet cell antibody. PICA is supposed to be pancreatic islet cell antibody, which is an autoantibody, destroys the beta cells of the pancreas and makes the person suffer from type 1 diabetes mellitus, and due to which insulin deficiency will take place and the person will be lifelong dependent on insulin for the regulation of glucose. Now, this is the normal architecture of the pancreas. Look at the architecture of the islets. The islets are completely destroyed and they are infiltrated by TH cells as well as the macrophages and the neutrophils. Once again, it is an inflammatory destruction of the pancreatic islet cells, okay, resulting in type 1 diabetes mellitus. So we will talk about the other diseases that are associated, where I don't have the pictorial presentation for that, but I will explain to you. One of them is autoimmune hemolytic anemia. We have already discussed that in the type 2 hypersensitivity, drug induced hemolytic anemia. I've already discussed with that. And yesterday I also discussed, when we discussed about the mimicking, or due to the epitopes being changed, change of epitope resulting in recognition of that epitope as the foreign antigen and autoimmune response. That also results in destruction of the RBCs because those epitopes are present on the RBC as band 3 protein. Okay. And drug induced hemolytic anemia, already discussed with you, where drug molecules act as haptens and those haptens will conjugate with the proteins of the RBC, and due to the conjugate of the proteins of the RBC, that will act as a foreign antigen, and the autoimmune response results in destruction of the RBC due to complement mediated lysis or C response. Then another type of hemolytic anemia is called as pernicious anemia. Pernicious anemia. And this pernicious anemia occurs in individuals who have vitamin B12 deficiency. Vitamin B12, that is cobalamin, vitamin B12 is called as cobalamin, is very, very important for the wellness of the RBC. Okay, RBCs will be healthy and they will finish their 120 days life cycle if vitamin B12 is there in your diet and you are taking it up through your intestine. Okay, and cobalamin is nothing but it is cobalt bound a peptide which is an amino acid residue. Okay, cobalamin. So that cobalt bound peptide which is constituting vitamin B12, okay, vitamin B12 is supposed to be absorbed from your intestine through the diet, whatever the vitamin B12 you take. And that protein molecule that is responsible for, okay, absorption of the cobalamin, that is vitamin B12, through the intestinal wall is called as intrinsic protein factor. Intrinsic protein factor. And that intrinsic protein factor is present on the membranes of the epithelial cells lining your intestine, where the absorption is taking place. And that intrinsic protein factor will be responsible for uptake of cobalamin, vitamin B12. And it is transferred through the blood to the bone marrow, where the RBCs are being produced and will be kept healthy because of the presence of cobalamin. Now let us presume that that intrinsic protein shows a minor change in the epitope of its amino acids, and that will result in formation of autoimmune antibodies which will destroy that intrinsic protein. And due to destruction of the intrinsic protein, the cobalamin, vitamin B12 will not be taken up by the intestine and that will result in the RBCs being produced less in number, and wellness of the RBCs will not be taken care. The person will suffer from deficiency of the RBC. That will result in anemia. Whenever the RBC deficiency takes place, the person will suffer from anemia. So that is one of the example for a hemolytic anemia. RBCs become very, very fragile. The absence of vitamin B12, they are very, very fragile. Minor change in the body pH kidney and the lung alveoli. So it is observed that the people will have auto-antibodies produced against their basement of the kidney, or it could be both, that is basement membrane of the kidney, that is glomerulus, as well as the alveolar region of the lung. Because of these autoantibodies reacting with the antigenic epitopes of the alveolar basement membrane, the person will suffer from destruction of the alveoli. And due to destruction of the alveoli, you know that alveoli are the regions where the oxygen is absorbed. Whatever you respire, that is facilitated in the alveolar region for the diffusion of that oxygen to the blood, and the blood carries the diffused oxygen from there to the heart. And you can imagine the surface is damaged there because of the autoantibodies causing destruction to the basement membrane of the alveolar region. That will incapacitate the person, and there will be respiratory insufficiency and suffer from anoxic injury and anoxia. The person will have deficiency of oxygen uptake, and that will result in ischemia or anoxic injury. The second one, the good posture syndrome about the autoantibodies against the basement membrane. And the basement membrane of the glomerulus, when it is destroyed because of the autoantibodies, the glomerulus, which is consisting of Bowman's capsule, will not be able to perform the proper function of filtration, and the kidney failure will take place, and the person has to lifelong depend on dialysis. So these are some of the examples as far as organ specific cellular destructions are concerned. Then had spoken about the second type under that, where the cellular destruction takes place much later, and the response is called as stimulatory hypersensitivity or stimulatory autoantibodies. I had discussed this in the Graves' disease. My explanation becomes easier now here. In the Graves' disease, which is an autoimmune disorder as well as type 5 hypersensitivity, I've already explained about this, where the autoantibodies are produced against the TSH receptor, thyroid stimulating hormone receptor, which trigger that cell to produce continuously thyroglobulin, resulting in hyperthyroidism in the beginning. Later, the same antibody will initiate ADCC response as well as complement mediated lysis of those thyroid follicle cells, resulting in destruction of the thyroid follicle, and causing hypothyroidism. Starts with hyperthyroidism, ends with hypothyroidism. That is one. Then the second one, the blocking antibodies as autoantibodies, causing a disease called as myasthenia gravis. Here in the picture they have shown the nerve ending, which is nothing but a synaptic vesicle, and that synaptic vesicle is responsible for the neurotransmission. neuroconduction, neurotransmission, and the neurotransmission is done to the surface area of the muscles. And I had told you that it is a voluntary action. You desire, you want to move your hand, means the action should take place on that particular hand, and you are directing that hand to move by the skeletal muscle action. The command goes to that skeletal muscle, and the voluntary command goes in such a way that the neurotransmitter such as acetylcholine diffuses in that area through the synaptic vesicle and comes and binds to the acetylcholine receptors of that region, causing that muscle to contract so that you can move that hand. It is your voluntary movement. You desired and you wanted, in your will, with willing, you did it. And that action is called as the non-autonomous action. You are doing it through your will, and which is through the acetylcholine neurotransmitter. Now let us presume that this normal action will take place provided that acetylcholine released by the synaptic vesicle goes and binds to the receptors on the muscle fibers, and that receptor is acetylcholine receptor. And the antibody produced, auto-antibody produced, will react on the acetylcholine receptors and goes and binds them. And because of the receptor being engaged already by the antibody, the receptor does not have the space for acetylcholine to bind. If the voluntary action takes place and the synaptic vesicles release the acetylcholine, that will fail to bind to the receptor and bring about that voluntary action, and due to which the person will suffer from voluntary movements. Say, for example, when you want to stand up, you give a command to your legs, and your brain will give command in such a way that you stand up by putting your legs on the firm ground and you stand up. But unfortunately, if that voluntary action also you can't do because of the autoantibodies binding to the acetylcholine receptors, you are bedridden. The person will get bedridden, and the person will be almost all the time will be lying on the bed because most of the voluntary actions he will not be able to. And I had told you the initial symptoms of myasthenia gravis is unable to close your eyelids. But if you have forcefully closed your eyelids by dragging it with your fingers, you will be unable to open it. That voluntary action also one person will lose. So this is the state of affairs of the autoimmune response against your own self cells. And one of them is stimulatory autoimmune response, another is inhibitory, where the antibodies cause inhibition of the transmission of nerve impulses. Okay, so this is as far as destruction. Now you know that when the antibodies are bound to the acetylcholine receptors, they will initiate the complement mediated lysis, and large number of receptors on the cells will be destroyed. In the bargain, the muscle cells also will be destroyed because of the mediated lysis or ADCC response. And you can imagine, as the person starts losing the muscle fibers, it will lead to wasting disease. Wasting disease means the person will start becoming thinner and thinner due to loss of muscles. Okay, loss of muscles. It is a progressive disease. One can control it. Nowadays, the therapies are available. Monoclonal antibodies are available, which can go and block these antibodies. All right, block these antibodies beforehand so that those antibodies do not go and bind to the receptor. Okay, acetylcholine receptor binding antibodies can be blocked using monoclonal antibody therapy, and you can decrease the impact of the myasthenia gravis in the person. It is suspected that the person is going to suffer from myasthenia gravis. Okay, so thank you very much. I'll stop here.